human body is always exposed to various kinds of harmful invaders like viruses, bacteria, fungi, protozoa, toxic substances, etc. Our skin is the first line of defense against disease-causing germs. These germs can rarely penetrate through tough outer layer of body. The body becomes vulnerable to attack only if these tissues are broken, for example, by a cut or an insect bite or a bigger animal bite. It has also been observed and proved that the persons who had been suffering from certain diseases like measles and mumps are not attacked in future by the pathogen of the same disease. Actually, they are attacked, but the body develops the ability to resist the attack by the same pathogen. This ability is known as immunity. Our today's lesson is all about this defensive mechanism of our body, that is, immunity.
Students, ਜਿਵੇਂ ਕਿ ਤੁਸੀਂ ਜਾਣਦੇ ਹੋ ਕਿ ਅਸੀਂ ਵੱਡੇ ਪੈਮਾਨੇ ਵਿੱਚ ਇਨਫੈਕਸ਼ੀਅਸ ਏਜੰਟਸ ਦੇ ਖਤਰਿਆਂ ਤੋਂ ਘਿਰੇ ਰਹਿੰਦੇ ਹਾਂ, ਪਰ ਇਹਨਾਂ ਵਿੱਚੋਂ ਕੁਝ ਹੀ ਹਨ ਜੋ ਬਿਮਾਰੀ ਦਾ ਕਾਰਨ ਬਣਦੇ ਹਨ, ਕਿਉਂਕਿ ਸਾਡਾ ਸਰੀਰ ਜ਼ਿਆਦਾਤਰ ਬਾਹਰਲੇ ਕਾਰਕਾਂ ਤੋਂ ਆਪਣਾ ਬਚਾਓ ਆਪ ਹੀ ਕਰ ਲੈਂਦਾ ਹੈ. Do you know that the word immunity arises from the Latin word immunis, which means exempt or freedom? Immunity refers to all the mechanisms used by the body for protection from external harmful agents. So, the ability of a body to fight against disease-causing organisms conferred by our immune system is called immunity. In other words, immunity refers to the resistance of a host to pathogens and their toxic products. The system of animal body which protects it from various infectious agents is known as immune system. ਸਾਡਾ immune system ਜੀਵਾਂ ਦੇ ਕਾਰਨ ਹੋਣ ਵਾਲੀਆਂ ਬਿਮਾਰੀਆਂ ਦੇ ਖਿਲਾਫ ਲੜਨ ਦੀ ਤਾਕਤ ਪ੍ਰਦਾਨ ਕਰਦਾ ਹੈ, ਜਿਸ ਨੂੰ immunity ਜਾਂ ਰੋਗ ਪ੍ਰਤੀਰੋਧਕ ਸਖਿਅਮਤਾ ਕਹਿੰਦੇ ਹਨ. Before going into details of this miraculous system, let us talk about the learning objectives of today's lesson.
Upon completion of this topic, the learners will be able to define immunity and susceptibility, list the types of immunity, differentiate between active immunity and passive immunity, explain vaccination and antitoxins, discuss the different ways by which immunity may develop.
ਅਸੀਂ ਆਪਣਾ ਅੱਜ ਦਾ ਪਾਠ ਵੱਖ-ਵੱਖ ਤਰ੍ਹਾਂ ਦੀ ਇਮਿਊਨਿਟੀ ਤੋਂ ਸ਼ੁਰੂ ਕਰਾਂਗੇ. ਬੁਨਿਆਦੀ ਤੌਰ ਤੇ ਵੇਖਿਆ ਜਾਏ ਤਾਂ ਇਮਿਊਨਿਟੀ ਮੁੱਖ ਰੂਪ ਵਿੱਚ ਦੋ ਤਰ੍ਹਾਂ ਦੀ ਹੁੰਦੀ ਹੈ, ਜਿਵੇਂ ਇਨਬੋਰਨ ਇਮਿਊਨਿਟੀ ਅਤੇ acquired immunity. Our body is able to fight against some of the disease-causing organisms. This ability of body to resist almost all types of foreign bodies or toxins which damage tissues and organs is called immunity. Immunity is developed by a complex system of different cells and tissues which all help to fight against germs. There is another term called susceptibility. Susceptibility is the vulnerability of a person to infectious agents. It is the opposite of immunity. I hope by now you are clear about today's topic, that is immunity. Let us discuss different types of immunity. Basically, there are two different major types of immunity. innate or inborn immunity, acquired or adaptive immunity. Let us talk about innate immunity. This type of immunity is inherited by the organism and protects it throughout its life. Thus, innate or inborn immunity comprises all those elements of protection which a newborn gets since his or her birth. This type of immunity is provided by various types of factors. One of them is presence of barriers in the form of hair, skin, inner organs, lining, etc. Let us see all types of barriers which provide innate immunity. These are physical barriers, physiological barriers, cytokine barriers, and cellular barriers. Physical barriers just act as checkposts to restrict the entry of organisms into the body, like skin. The outer tough layer of the skin prevents entry of bacteria. This layer consists of dead cells and is filled with a protein called keratin, which makes the skin tough and safe. Mucous coating of the epithelium, lining of the respiratory, gastrointestinal and urogenital tracts traps microorganisms and immobilize them, thus preventing their entry into our blood of deep skin. Physiological barriers of our body like temperature, pH, some body secretions prevent growth of microorganisms in our body. Dermis of skin is a thicker inner layer which contains sebaceous glands associated with hair follicles, produce sebum which consists of lactic and fatty acids and maintain pH of 3 to 5. Such a low pH is not suitable for growth of microorganisms. Normal body temperature also inhibits growth of most microorganisms. Acid produced in our stomach kills bacteria. Airwax produced traps dust particles. Saliva in the mouth and tears from eyes also prevent microbial growth. Now let us discuss cytokine barriers.
By cytokine barriers. Whenever the skin is broken due to some minor injury, that area becomes inflamed and shows 4 symptoms: redness, pain, swelling, and heat. Let me tell you why it all happens and does it provide any type of protection? During injury, a ruptured capillary releases bradykinin. This molecule stimulates receptors of nerve impulse, result in sensation of pain. Along with this, mast cells are stimulated to release histamine, which together with bradykinin causes capillaries to dilate. The enlarged capillaries cause the skin to redden. Rise in temperature reduces the number of invading microbes. A rise in temperature reduces the number of invading microbes and increases phagocytic action by white blood cells. This phagocytic action takes place with the help of some cellular factors. Cellular barriers. Certain types of white blood cells like neutrophils and monocytes present in our blood can engulf microbes which somehow manage to enter after crossing physical and physiological barriers. This action of engulfing bacteria by these cells is known as phagocytosis, which you have studied many times. We know that neutrophils and monocytes have amoeba-like body. By forming a vesicle, these cells engulf bacteria and destroy them by releasing hydrolytic enzymes. These are the factors which usually depend on our genetic constitution and thus are discussed as innate or inborn ability. Next is acquired or adaptive immunity. This immunity is acquired by an individual during his lifetime. This immunity is developed by an individual in response to a disease caused by infection of microbes. This protects body from invasion of microorganisms. Thus, it relieves the victim of infectious disease. Two distinct types of lymphocytes known as T lymphocytes and B lymphocyte cells form memory cells which are the basis of acquired immunity. Lymphocytes are made in liver of fetus and bone marrow of adults.
Students, you know acquired immunity is again of two types: antibody mediated immunity or humoral immunity and cell mediated immunity. Let us discuss antibody mediated immunity. This type of immunity is achieved by chemicals. No cells are directly involved. There is a great role of first time response, that is primary response, in this type of immunity. It is based on the fact that whenever our body encounters a pathogen for the first time, it produces a response known as primary response, which is of low intensity. Subsequent encounters with the same pathogen causes a highly intensified secondary response. This secondary response results in production of certain protective chemicals known as antibodies. Antibodies are produced by B lymphocytes in our body. B cells are produced in the bone marrow and are distributed throughout the body in the lymph nodes. These B cells respond to foreign antigens of pathogen by producing specific antibodies. These cells do not fight directly. This happens because our body develops some memory cells during the first attack.
When a pathogen tries to invade through a body for the first time, B lymphocytes get activated. They start secreting antibodies specific for the invaded pathogen. When the infection is over, most of B lymphocytes die. This sequence of events is known as primary immune response. Whenever there is an attack by the same pathogen, body reacts more quickly as some of its cells behave like memory cells. They are able to remember this pathogen and divide into large number of B cells, all capable of secreting antibodies. This response is called secondary immune response. It is much quick and more effective than the primary response. Let's see the structure of an antibody. Here you see the structure of an antibody. Each antibody is made up of 4 peptide chains. 2 of them are smaller in size and are known as light chains, whereas the other 2 are longer. These longer ones are known as heavy chains. Because of these 2 types of chains, each antibody is represented by H2L2. H stands for heavy and L stands for light. The 4 peptide chains are held together by disulfide bonds to form Y-shaped molecule.
Now let us discuss about another type of acquired immunity, that is cell-mediated immunity. As the name suggests, it involves specialized cells called T lymphocytes which directly attack the pathogenic organisms. T lymphocytes present in our blood recognize self and non-self, and these cells act accordingly. About their role in immunity, it can be explained easily by remembering the case of an organ transplant. Huge research is done to find a suitable donor. Why is it not possible to take organ just from anybody and transplant it in the patient? What do doctors check while selecting suitable donor? They check response of T lymphocytes, which is done by tissue matching, blood group matching, etc. Similarly, activated T lymphocytes attack and kill whenever a foreign body enters. The body is able to differentiate self and non-self, and cell-mediated immune response is responsible for graft rejection. Like B cells, T cells respond to specific antigens. Some of the activated T cells become memory cells and remain in the body. They remain ready to give a secondary response if pathogens attack again. Some of the T cells are of three types on the basis of function they perform. Some of them become helper cells. They stimulate B cells to divide and produce antibodies. Killer T cells attack infected body cells. Another very important function is performed by a group of T cells which provide a sort of safety cutout mechanism in our body. You know, when our immune system works excessively, then these T cells ask it to shut down, because if the body continued to make antibodies even when not required, it could damage the body. This group of cells, which suppresses the whole immune system to protect own body cells from the attack, are known as suppressor T cells. Immunity has also been classified on the basis of active and passive response of our body.
Active immunity and passive immunity. When a host is exposed to antigens in the form of live or dead microbes, antibodies are produced in host body. This takes longer times but gives full active response. Injecting microbes deliberately during immunization also develop this type of response. This type of immunity is active immunity. During this active response, the body's own cells produce antibodies. During passive immunity, ready-made antibodies are injected directly to protect the body against foreign agents. Passive immunity provides immediate relief, whereas active immunity requires time for the formation of antibodies, and relief is after a long period. There are other differences also. Passive immunity is not long-lasting, whereas active immunity is long-lasting. Passive immunity may cause reaction, whereas active immunity has no side effects.
You know, mother's milk is considered very beneficial as it provides passive immunity. The yellowish fluid, colostrum, secreted by mother during initial days of lactation contains antibodies which protect the infant properly by the age of 3 months. Active immunity is long-lasting as compared to passive immunity. ਇੱਥੇ ਸਵਾਲ ਇਹ ਉੱਠਦਾ ਹੈ ਕਿ ਉਹ ਕਿਹੜੇ ਸੈਲਸ ਹਨ ਜੋ ਇਸ ਤਰ੍ਹਾਂ ਦੀ ਯਾਦਾਸ਼ਤ ਨੂੰ ਬਣਾਏ ਰੱਖਣ ਵਿੱਚ ਸਾਡੀ ਮਦਦ ਕਰਦੇ ਹਨ। ਇਮਿਊਨਿਟੀ ਬਿਮਾਰੀਆਂ ਦੇ ਖਿਲਾਫ ਲੜਨ ਦੀ ਸ਼ਕਤੀ ਹੈ। ਸੋ ਬੱਚਿਓ, ਇਹ ਸੀ ਵੱਖ-ਵੱਖ ਤਰ੍ਹਾਂ ਦੀ ਇਮਿਊਨਿਟੀ ਬਾਰੇ ਜਾਣਕਾਰੀ ਜਿਸ ਨੂੰ ਮਨੁੱਖ ਆਪਣੇ ਅੰਦਰ ਵਿਕਸਿਤ ਕਰ ਸਕਦਾ ਹੈ। ਪਰ ਇੱਥੇ ਸਵਾਲ ਇਹ ਹੈ ਕਿ ਇਹ ਕਿਸ ਤਰ੍ਹਾਂ ਵਿਕਸਿਤ ਹੁੰਦੀ ਹੈ। ਇਸ ਦੇ ਬਹੁਤ ਸਾਰੇ ਤਰੀਕੇ ਹਨ ਜੋ ਇੰਜ ਹਨ। Immunity through infection. When first time a person gets any infection, the disease is definitely expressed in his body. Subsequent infection by the same microorganisms may not cause the disease. This is the reason we usually say that a newborn child will get more infection as compared to elders. Why does it happen so? Let us work out. In the case of first infection, body reacts but antibody formation is very slow. With the result, the pathogens get time to multiply and spread throughout the body and disease appears. A stage reaches where the rate of antibody formation picks up and the disease disappears. Cells capable of producing antibodies persist in the body as memory cells for a long time, maybe for lifetime after the first infection. Whenever there is a second attack by the same pathogen, these memory cells soon become active, produce antibodies of the same type. Some diseases like measles, mumps, smallpox and polio once got produce a lifelong immunity. For diphtheria and typhoid, immunity lasts for a few years, but not for the whole life. Another way is the immunity through vaccination. ਬਿਨਾਂ ਲਾਗ ਦੀ ਬਿਮਾਰੀ ਵਿਰੁੱਧ immunity ਨੂੰ ਵਧਾਉਣ ਦਾ ਸਭ ਤੋਂ ਵਧੀਆ ਤਰੀਕਾ ਹੈ vaccination. Vaccination is a technique to develop immunity without infection. Weakened or dead pathogens are injected into a person. Weak or dead pathogens in the vaccine are unable to cause the disease, but they are sufficient to stimulate the formation of antibodies in the host body and generate memory B and T cells, so that whenever there is attack by the same pathogens, the existing memory T or B cells recognize the pathogen and attack it with massive production of lymphocytes and antibodies. Thirdly, now we will discuss immunity through antitoxins. Let me explain what are antitoxins.
Antitoxins. Those antibodies which can neutralize toxins produced in the body due to some pathogenic attack are known as antitoxins. Antitoxins are prepared artificially also. Students, you know how toxins against snake bite is prepared? Snake venom is injected in low dose to an animal, usually a horse. The animal develops antibodies against the snake venom in its blood. This blood containing antibodies against antigen is drawn and allowed to clot. The serum, which is left behind after clotting, contains antibodies against snake venom. This serum is injected into a person bitten by a snake to neutralize the snake venom.
We are all surrounded by many types of germs that are capable of causing diseases if they enter into our body. But our body is able to defend itself against most of foreign agents by its ability to fight against these disease-causing organisms, and this ability of the body is called immunity. But sometimes these germs resist the efforts of our body to reside and multiply inside our body, thus causing diseases. These germs also try to go from one host to another host, that is what we call spread of infection. There is a system to provide us protection by fighting out these invading germs. This system is known as immune system. Through this system, we get immunity. The ability of the immune system to distinguish between invading organisms and the tissues of its own body is another way to define immunity. This concept of self and non-self is important in determining whether the immune system keeps the body healthy or whether it is overcome by infection. There is another term called susceptibility. Susceptibility is the vulnerability of a person to infectious agents. Immunity may be innate or inborn, and acquired or adaptive. Inborn immunity is inherited by the organisms from parents and it protects it throughout life. Acquired immunity is developed in an individual in response to a disease caused by microbes. There are certain natural barriers in our body which protect us from these invading germs. For example, nasal hair. They filter the air that is breathed in. Skin. Upper layer which is made up of a tough, impermeable layer of cells. Certain physiological factors of our body like temperature, pH, some body secretions also prevent growth of microorganisms in our body. Mucous membrane which lines up nose, eyes, mouth, etc. produces a sticky fluid. This secretion traps germs and stops them from attacking our body. Certain fluids such as sweat, saliva and tears contain chemicals which inhibit the growth of germs in our body. Immunity may also develop through vaccination. Weakened or dead pathogens injected through vaccination stimulated formation of antibodies to develop immunity. Antitoxins are antibodies that neutralize toxins. These are prepared artificially and are used to counter at snake bite. So students, by now it is clear what is an immune system and what are the different types of immunities, how they function to affect immunity in a living organism. Now, let us quickly check your knowledge with some short questions as to what you have grasped. Please be attentive. Here are some questions. The first question is, what is the technique of developing immunity called? The answer is, immunity can be developed through vaccination. The next question is, name the first disease which was eliminated by vaccination method. Answer, yes, vaccination method was first time used to eradicate smallpox. My next question is, what is the name given to antibodies that neutralize the toxins produced in the body? And the answer is, those antibodies are known as antitoxins. Next question. Out of passive and active immunity, which one provides immediate relief? The answer is, passive immunity provides immediate relief. The next question is, how a newborn baby gets protection against germs? Answer, because it has temporary protection by antibodies received from mother's blood before birth and mother's milk after birth. The next question is, by how many methods immunity can be achieved? Answer, immunity can be achieved by these methods. Immunity through infection, immunity through vaccination, and immunity through toxins. My next question is, name the main cells of the immune system. The answer is lymphocytes. The next question is, what type of immune system is formed by B lymphocytes? The answer is AMIS, which is antibody mediated immune system, is formed by B lymphocytes. The next question is, what kind of immune system is formed by T lymphocytes? And the answer is CMIS, that is cell mediated immune system is formed by T lymphocytes. The next question is, what is the role of suppressor T cells? Answer: to check excessive response of immune system means to avoid wastage of body resources. These cells stop immune system to work further. Well students, in this class we have studied about the types of immune systems and how they function in our body. Thanks for your attention. Looking forward to meet you in next class. Till then, goodbye.